Hello, everyone, and welcome to today's broadcast, Outer Membrane Remodeling of Attenuated Salmonella to Create a Mucosal Vaccine Against Acinetobacter baumannii, presented by Dr. James Galen, Professor, Center for Vaccine Development, University of Maryland, Baltimore. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Galen. I will now turn the presentation over to him.
Well, thank you very much for the opportunity to speak to you all this afternoon. We are going to be talking about a concept called carrier vaccines. Carrier vaccines are typically vaccines that are engineered from a bacteria to express foreign antigens from unrelated human pathogens expressed in this vaccine strain. Typically, we prefer to use vaccine strains derived from human pathogens and not human commensals, because human pathogens are exquisitely adapted for invasion into mucosal tissue and triggering immune responses, where commensals typically are not adapted to do those things. So the bacteria, the human pathogen that we've chosen to work with is Salmonella typhi, which is the causative agent of typhoid fever. And we have engineered Salmonella typhi to be an attenuated vaccine strain, and we have further adapted that to become a carrier vaccine. So what are the key requirements for developing bacterial live carrier vaccines? Well, first of all, since our vaccines are intended for human vaccination, they have to be clinically acceptable. In other words, they have to be safe and immunogenic. And so we have, we started out this project many years ago with CVD908-HTRA, which has been proven safe and immunogenic in phase 2 human clinical trials. For reasons I will try to get into here, CVD908-HTRA was not a good vaccine strain to adapt to become a carrier vaccine. So I re-engineered a strain called CVD910 with attenuation comparable to the clinically acceptable CVD908-HTRA. The second thing we need is we need efficient foreign antigen expression. So over the years, we have developed patented non-antibiotic plasmid selection technology. Obviously, we're not going to want to use plasmids that require antibiotic selection when we're trying to target a human pathogen for which use of antibiotics has caused problems in the first place. Also, we have over the years developed patented antigen export technology, which is going to be the primary focus of the talk today. And last, but certainly not least, we need acceptable preclinical animal models for testing and refining the immunogenicity of vaccine strains. This is really important. If you spend years developing engineered strains, but you don't have an animal challenge model, it's going to be very hard for you to get into the clinic. So you need acceptable animal models, and we have 2 at this point. We have a murine mouse mucosal, i.e. intranasal immunization model, and we have actually expanded this intranasal model to cover non-human primates. And we'll go through some of this technology and the animal models as we proceed in this talk. So I got to say, the road to developing a live oral carrier vaccine has been very, very, very long. And it has also been quite a ride. When I first got into this field a couple of decades ago now, decades, there really weren't that many tools available. So as the years have gone by, we've had to create a lot of the tools that we've needed. So first thing I want to talk about is if we're talking about carrier vaccines, right? So we need to express these foreign antigens in a way that the immune system can see these antigens and mount the appropriate immune response against these foreign antigens. Okay, so what are the factors affecting this immunogenicity of foreign antigens? It's very important to understand that the carrier vaccine typically does not benefit from expression of these foreign antigens. For example, we're going to discuss in this presentation development of an anthrax vaccine where we target protective antigen PA83 from anthrax toxin. So we're going to express PA83 in our carrier vaccine. Now, if Salmonella Typhi was supposed to be expressing PA83 naturally, it would already be doing so, but it doesn't. And therefore, when we come in and engineer hyperexpression of this antigen, we typically increase the metabolic burden on the strain, which is already attenuated. And if we inappropriately overexpress these foreign antigens, we end up with a vaccine strain which is so attenuated that it's very safe in clinical trials, but it has no immunogenicity. And we'll talk about this. And the last thing that it's taken a few years to figure out is that exported or surface expressed foreign antigens are more immunogenic than cytoplasmically expressed foreign antigens, which are expressed inside the carrier vaccine strain. So we want these antigens to be on the surface and not inside the organism, okay? All right, so let's look at attenuation here. Over the years, a number of attenuated Salmonella Typhi vaccine strains have been developed just as vaccine strains, not as carrier vaccines, okay? And what we found over the years is that over-attenuated strains, okay, are poorly immunogenic. So if the strain is too weak, then it doesn't really reach the appropriate immune inductive sites, and you get a very safe vaccine, which is not immunogenic. Okay, insufficiently attenuated vaccine strains are extremely immunogenic, but they're reactogenic, so they make you feel that you get clinical symptoms. Unacceptable clinical reactogenicity is the way we like to call it, okay? So the key is to achieve the right balance between attenuation and immunogenicity. And this is shown very clearly in the next slide, which brings up another point that I want to discuss. Okay, so balanced foreign antigen expression will enhance the immunogenicity of a live carrier vaccine. So the top panel here, A, B, and C, you can see a vaccine strain which is very, very, very reactogenic is also immunogenic. But it's too reactogenic, so the safety is unacceptable, okay? When you engineer, when you attenuate the vaccine strain so that it's not reactogenic, it's not immunogenic, okay? And so what you have to do is you have to approach just the right balance between safety and immunogenicity, okay? But this is a problem because the perfect vaccine strain is already metabolically balanced. So when we come in with foreign antigen expression, we destroy that balance. And that's why developing carrier vaccines has been so difficult. So A, B, and C address how to make a vaccine strain. But what if we want to make a carrier vaccine out of that beautifully balanced vaccine strain? Okay. So what I've figured out over the years is you want to start with a vaccine strain which is slightly reactogenic. If you overexpress on extra high levels of foreign antigen, it becomes over attenuated, and so it's non-reactogenic, but it's also not immunogenic. So you have to adjust antigen expression just right so that you get a balance between safety and immunogenicity against both the carrier vaccine and the foreign antigen. And this is tough. This is hard to do. Okay? So our vaccine strain that we started with, remember I said I had to go back and re-engineer a vaccine strain because our original vaccine strain, CVD908HTRA, was perfectly balanced, but then when we started expressing foreign antigens, it became over attenuated. So I went back and engineered a strain called CVD910. Okay, it's engineered from the same wild type typhoid strain, TY2, as CVD908HTRA came from, and also the currently licensed typhoid vaccine strain, TY21A, comes from the parent strain, TY2. Okay, CVD910 contains precise genetic deletions in chromosomal loci, GUA-BA and HTRA. So this is important because what this means is our vaccine strains are attenuated metabolically, but any known or unknown virulence factors that are present in Typhi are still in our vaccine strains. So these vaccine strains go in after immunization of a host. They go in ready to do what they've always done, but because of the metabolic attenuation, they run out of gas. And so therefore, you don't see a disease state after immunization because that would be bad for a vaccine. Okay? So the attenuation of CVD910 was confirmed in biopsy mice according to standard LD50 assays, and our LD50 lethal dose 50 assay for CVD910 was equivalent to CVD908HTRA. So therefore, we feel that CVD910 is a clinically acceptable strain, but that, of course, has to be demonstrated in clinical trials, phase 1 clinical trials. Okay, so this slide is going to frame the rest of the presentation today, okay? So we have a vaccine strain, but we now have to get all of the antigen expression technology into place, the appropriate antigen expression technology, in order to develop a carrier vaccine from CVD910, the vaccine strain, the parent vaccine strain. So what do you need to develop? What kind of expression technology do you need? Okay, well, first of all, plasmids are going to fit in very nicely into development of carrier vaccines. And plasmids are notoriously unstable in Salmonella typhi. So you need a plasmid partitioning system to guarantee stable inheritance of these expression plasmids into dividing daughter cells of CVD910. If you lose the plasmid in vivo, you lose the ability to make foreign antigen. So you may get a really good antibodies response or an immune response against the live vector of the carrier vaccine, but you won't get any immune recognition of the foreign antigen because the plasmid's gone, okay? The second thing, which we've alluded to, is we need an antigen export system so that we get high-level expression of vaccine antigens on the outside of the cell. The third thing, which I got dragged kicking and screaming into the development of, was a non-antibiotic plasmid selection system. Because in the good old days, when things were very simple, in the laboratory, you can have plasmids and your E. coli strains, and you can select them with antibiotics, and every cell has the plasmid, and it's lovely, and life is grand. Well, you can't use antibiotics in an oral vaccine intended for human administration especially if you're trying to combat an antibiotic-resistant pathogen. So you have to develop a plasmid selection system that no longer uses antibiotics. And that took that took some time. And then last but not least, we've had to also develop chromosomal expression systems, because we realized if we want to express more than one antigen in a single carrier vaccine, you're not going to be able to put all of those on a plasmid, because the plasmid would be huge and it's unstable, and then it'll be disappearing. And again, you end up with non-immunogenic, beautifully engineered carrier vaccines, but they don't work. Okay, so let's look at plasmid partitioning and antigen export. Let's look at that in a little bit more detail. All right. Okay, so we have several hypotheses for engineering multivalent Salmonella and Typhi carrier vaccines. Okay, so this is my great hypothesis part one. Okay, live vectors derive little or no benefit from expression of foreign antigens. Okay, and in fact, these foreign antigens may be harmful if they're expressed intracellularly, because they may block anabolic functions or a number of other ways. Not to mention the fact that if you have high-level expression of intracellular antigens, if the level of expression is too high, then they start to improperly fold, and then they form inclusion bodies, and inclusion bodies will not present the right, if necessary, conformationally specific antigens, and you won't get that, and therefore, again, you have a vaccine that's beautifully engineered, but it doesn't work. Okay, so what we ended up deciding to do after a long time was we ended up first going with a low copy number expression plasmid. Okay, so like I referred to, the good old days, you know, we used to use high copy number puck plasmids, and life was just grand. You throw in a little IPTG and bang, you've got tons of antigen. You can't do that in a live vector system because of the balance that we're trying to achieve here. So we actually found that when we appropriately expressed antigen at the right levels from low copy number plasmids, we actually ended up getting superior immune responses. Okay, so we need a low copy number expression plasmid. Ours was derived from a plasmid called SC101. It has an ORI101 replicon. It's about 5 copies per chromosomal equivalent. Next thing we need is plasmids are not chromosomes, okay? And so for the most part, plasmids will, when a daughter cell elongates and divides, the plasmids will be passively inherited just by random sequestering into daughter cells. That's not good enough for live carrier vaccines. So we needed a stable inheritance system called PAR-A, and I would really love to get into the details on this. It's fabulous stuff, but I don't have the time here. But anyway, this partitioning system pushes copies of the plasmid into daughter cells. It's fabulous stuff. Okay. Again, we need an inducible antigen expression system. Full bore, always on, constitutive expression in a carrier vaccine. Remember, we're trying to get balance, okay? Constitutive expression, it does not work. It over attenuates the strain, you get nothing. Okay, so we need an inducible antigen expression system. So we have chosen the OMP-C promoter, okay, which is osmotically controlled. So when the osmolarity is high, it turns on. When the osmolarity is low, it turns off, okay? And we also have this antigen export technology, which is called CLI-A, okay? And as I referred to earlier in the presentation, we're focusing on PA83, which is protective antigen 83 kilodalton from anthrax toxin. That project has allowed us to develop a lot of this technology, all right? But I gotta show you this slide because this is really cool, okay? So we started working with a protein called CLI-A. CLI-A mediates foreign antigen export, okay? And it can mediate either surface expression or outright secretion by outer membrane vesicles into the supernatant extracellular medium. All right, so when I first developed this export technology, I wanted to see if Cli-A could actually do this. And so I focused on a protein called SAC-B. And for some of you that have used SAC-B in enteric bacteria engineering, SAC-B makes an enzyme called levan sucrase. Levan sucrase has this remarkable ability to take sucrose, it cleaves it into glucose, and leaves fructose. But because levan sucrase doesn't have anything else to do, it doesn't have any meetings to go to or anything, so it takes the fructose and links it together, because it doesn't have anything else to do, and produces this long polymer called levan, okay? And levan almost looks like a capsule. It's like this gloppy mess, all right? If SAC-B makes levan intracellularly, you've got dead bugs. But if SAC-B ends up on the outside of the cell, then if it's exposed to sucrose, it makes levan, and the levan coats the outside of the cell. So what we did was we made a genetic fusion of Cli-A to SAC-B to test the hypothesis that Cli-A could move a large foreign antigen domain out to the surface of Cli-A. And then we plated these transformed cells onto 16% sucrose. Okay, so on the left, you've got bacteria plated on a 16% sucrose plate where they express Cli-A, but it's not fused to SAC-B. Okay, so the bacteria grow fine. Now, if Cli-A was not able to efficiently move SAC-B out to the surface, when I plate cells containing Cli-A SAC-B, you should get nothing growing on the plate. But that's not what happened, okay? And so you got these cells that are making a huge amount of levan. And in fact, if you look carefully at the plate, I bumped the right side of the plate before I took the picture, and the levan goo flowed over to the right. I mean, we're talking about a tremendous amount of levan here. So this showed me very clearly that Cli-A is indeed able to transport foreign antigen domains to the surface quite efficiently, okay? And so when I saw that plate, I thought, ha, we're in business, all right? But NIH doesn't care about expression of Cli-A and SAC-B on the surface of cells, so we had to like do something relevant here. So what we did, I keep referring back to PA83. We ended up making a vaccine against anthrax, targeting protective antigen from anthrax toxin. And in this slide, in panel A, you can see Salmonella Typhi. This is an electron micrograph, okay? Gold labeling. The gold labeling is targeting PA83, okay? And you can see Cli-A PA83 ends up on the outside of the cell exactly as intended, okay? And yet, when you look at gold labeling of Salmonella Typhi carrier vaccines expressing cytoplasmic PA83, obviously, as expected, it doesn't end up on the surface, okay? So Cli-A is not only able to move SAC-B out to the surface, but it's able to also move protective antigen 83, which is a much more relevant antigen for the real world. It moves that out to the surface as well. Now, remember I said if you've got a carrier vaccine and you're all dressed up, but you don't have anywhere to go, then you've got some nice engineering, but it doesn't really mean anything unless you test it in a clinically relevant animal model, okay? And so we did a lot of preliminary testing in mice, and we found that antibody responses against PA83 were they were excellent, okay? But because we had access to some primates from NIH, we conducted a non-human primate intranasal model of an immunogenicity study. And you notice that this study went on for 130 days. It was a very long experiment, but it worked really well. And what we ended up finding out was that when you immunize cynomolgus monkeys intranasally on days 0 and 14 with our carrier vaccine expressing CLY-A-PA83, and you look for toxin neutralizing antibody, okay, which is on the y-axis here, after 2 doses, you get nothing, okay? And so we knew ahead of time that the best way to produce excellent immune responses was with heterologous prime boosting, where your antigen, PA83, is delivered to a host in 2 different forms, okay? So heterologous prime boosting in this particular setting was carried out by intranasal mucosal immunization with the live vector, the carrier vaccine, and parenteral boosting with purified PA83. All right, so the blue arrows show that after 2 intranasal doses of carrier vaccine, no obvious toxin neutralizing antibody was detected. Okay, but then on day 100, we boosted parenterally with alum adjuvanted PA83, and the immune responses were excellent after that. And what's really interesting is if you look at the orange inverted triangles, that's the immune response against, let's see, let me get this straight. That's the immune response against intracellularly expressed PA83. But if you look at the blue curve here, that's the antibody response against extracellularly surface expressed P83. So again, this slide demonstrates in a non-human primate model that surface expressed proteins are more immunogenic than cytoplasmically expressed proteins. Okay? All right, so now we're coming back to this same slide that frames the rest of this talk. Now let's look at the non-antibiotic plasmid selection system. This was the project that I got dragged into kicking and screaming because this was going to be tough. How do you select for a plasmid without using antibiotics? Okay, and what we ended up choosing was a gene that called SSB, that encodes single-stranded binding protein. And single-stranded binding protein is essential for all living cells. If you don't make SSB, you're dead, okay? And if you look at this slide, because I always there's 2 things that I always have to do in a presentation. I always have to present a plasmid, which calms me down and makes me more confident. And I always have to present a crystal structure slide because it's just cool. All right, so if you look at this crystal structure slide here, you see this little ribbon of white that goes all through these tetramers of SSB. That's the single-stranded binding protein. So SSB binds to single-stranded DNA and keeps it single-stranded, which is very, very important for DNA replication, recombination, and repair. And so if you don't make SSB, then you can't do DNA replication, recombination, and repair, and you don't have life. And I don't have a chance to go through the details of this, but you can imagine that if we tried to delete SSB from the chromosome of our Typhi vaccine strain, and then put that SSB on a plasmid, and then introduce that plasmid into SSB minus strains, you can see where this could get a little bit complicated. And it was, in fact, very complicated, but it actually did work, and we got the system working correctly. And so we were then able to test this to find out, what does it do? Does it actually maintain plasmids. And so we published this paper back in, I think, 2010. Wow, time flies when you're having fun. And sure enough, we got some really very, very interesting data. Okay, now this was back in 2010, so we were still using 908-HTRA as the attenuated vaccine strain. Okay, and what we did was we did intranasal immunization of mice on days 0 and 14. Again, intramuscular boost of mice on day 42 with purified PA83. So this is a heterologous prime boost strategy. And we measured humoral responses against PA83. Okay? So in this table here, let's just look at CBD 908-HTRA carrying our expression plasmid. Okay, and you notice the expression plasmid has a copy number of about 15. The foreign antigen being expressed is CLY-APA83. There's no SSB stabilization. Okay, on day 41, right before the boost, okay, the humoral immune response was 80 in terms of ELISA units. Okay, on day 48, 1 week after the boost, it went up to about 14,000. Okay, now notice that when we bring in the SSB stabilization system on a 15 per copy plasmid, okay, day 41 pre-boost was 25, and the day 48 1 week post-boost dropped tenfold. And I thought, oh man, okay, so on the surface of it, it didn't work, right? All right, however, I realized that, and I sort of saw this coming into this, once you take a multi-copy plasmid and you stabilize it so that the carrier vaccine has to keep that plasmid, okay? The good news is that if you have a good stabilization system, the plasmids are stabilized. But the bad news is if those plasmids are encoding an antigen at too high a level and you get metabolic burden, your antibody responses will drop. So we went to 908-HTRA SSB with 5 copies per plasmid. CLY-APA83 fusion, SSB. Notice on day 41 before we get to the boost, it's already gone up to 2,300. And then 1 week post-boost, it goes to 156,000. And then when we went to a less attenuated background strain, here again, metabolic burden. This is a healthier strain. 908 is healthier, less attenuated than 908-HTRA. Remember that balance we talked about, okay? Day 41 pre-boost was now almost 5,000, but then after 1 week after post-boost, it went up to 500,000. So when I saw these data, I realized SSB is the way to go for antigen expression from a low copy number stabilized plasmid. Okay. Last but not least, chromosomal expression systems. Okay. The second part of the hypothesis here, this is hypothesis for engineering immunogenic multivalent salmonella typhi carrier vaccines, part 2. We hypothesize that it's unlikely that immunogenic and protective multivalent live vector vaccines expressing more than one foreign antigen, using only plasmids, you're not going to get away with it. They're going to be too big and they're going to be unstable. Okay? All right. So therefore, if you can't get them all on a plasmid, what do? And the answer is you start integrating some of these antigens into the chromosome. Okay? However, because you've integrated into the chromosome, your gene dosage drops. So if you're on a 5 copy per plasmid, okay, you've got 5 copies of your foreign antigen that are independently expressing. If you go to the chromosome, you only have 1 copy. So therefore, it's likely that you're going to have multiple integrations into the chromosome to get the antigen expression that you want. Okay? All right. So I'm just going to go through this very briefly. This was a concept project that we wanted to find out, well, if you use, instead of a foreign antigen, if you use green fluorescent protein as your reporter, is it true that when you integrate into multiple loci, you get an additive effect and you get higher levels of expression, okay? And so we integrated into the Guabier locus, all right, and we got a mean fluorescence of 32. We integrated it into the HTRA locus, and we got a mean fluorescence of 24, but we integrated it into both loci. Indeed, we saw an additive effect, and it went up to 48. Okay, so this encouraged us that multiple integrations sites in the chromosome would be the way to express appropriate levels of a single foreign antigen cassette. And so with CBD 910, we come up with this plausible design for a carrier vaccine expressing up to 4 antigens. If each one of these antigens only needs a little bit of expression, we can integrate it into Guabier, HTRA, RPOS, or a plasmid. And you notice the SSB stabilizing that plasmid, and SSB is knocked out of the chromosome. Okay? All right. Now, I had to throw this slide in because I've never seen this before. It's really cool. We did not know this before we started looking at it, but we started, because, what if I need 4 chromosomal loci? I have Guabier, HTRA, and RPOS, but what if I needed yet one more integration site that I knew inactivation of that locus wasn't going to further attenuate the strain? And we started looking at the Cli-A locus. And so here again, we've got an OMP-C cassette driving GFP expression integrated into the Cli-A locus, where we've kept the promoter for Cli-A intact. This is the strategy that we've used with all of our integrations into the chromosome. The target locus of the chromosome, we always keep the promoter. So if we integrate into Guabier, we keep the Guabier promoter. If we integrate into HTRA, we keep the HTRA promoter. In this case, we kept the Cli-A promoter. And this was very, very interesting. In line 1 here, liquid 25 millimolar sodium chloride. Okay, so that's the OMP-C promoter is theoretically turned off. Okay, we got a mean fluorescence of 33. When we grew the strain in liquid and we induced with 150 millimolar sodium chloride, the mean fluorescence went up to 76, but I expected it to be higher than that. And we started to think about it. So we took the same strain and we plated it on solid liquid, a solid LB medium with low salt, which turns out to turn on the Cli-A promoter, which we didn't know this until we ran this experiment, and the fluorescence jumped to 131. That was a big surprise. And then when we plated on solid media with 150 millimolar sodium chloride, in other words, inducing conditions where both the Cli-A promoter and the OMP-C promoter are on, we got very high fluorescence of almost 200. So this showed me that Cli-A could be another locus for integration, but a very special locus where Cli-A promoters would only turn on upon solid phase induction. In other words, let's say colonization of the host, okay? All right, so why am I interested in all these exotic promoters here, okay? What we really don't want when we immunize a host is we don't want all the antigens to be expressed at the same time, because here again, we go back to this balance. If we over-attenuate the strain due to heavy antigenic expression, we have a strain that's not immunogenic. What we'd really like to do is have the antigen induced at various phases of colonization and that would be expected in the immunization process, so that antigens get expressed a little at a time as the vaccine strain starts to take hold and starts to reach inductive sites. So you can see after oral immunization, we can envision a situation where when the organism reaches the gastrointestinal tract, the stomach, you get osmolarity induced, okay, for antigen 1. If you had antigen 2 integrated into the HTRA locus, you may also have a heat shock response when it hits that acid barrier. Okay, so you can see more antigen being expressed. Okay, as the live vector, as the carrier vaccine makes its way into the gastrointestinal tract, you may get some temporary colonization. Solid phase induction, Cli-A may be induced, maybe making more antigen 2. Okay, we're gradually introducing foreign antigen expression geared towards the metabolism of the live vector itself. That's the key that we're trying to achieve here. Okay, after colonization, there may be an exponential phase where the cells really start to grow. That induces the Guabier locus, and you may start to express, let's say, foreign antigen 3. You see this gradual introduction of antigen expression based on environmentally relevant signals, okay? And then finally, the organisms may reach some sort of stationary phase. That'll trigger the RPOS promoter, a little bit more antigen 3 in this example, okay? So it's gradual expression. Okay, so to make a long story short, eventually the biodefense funding kind of dried up, and we were not able to take our anthrax vaccine into clinical trials. So the bad news was we never really got to be able to test this in a clinical trial, but the good news was that the technology that was developed for this carrier vaccine is applicable to any pathogen, okay? And so we decided to move on and target Acinetobacter baumannii, which the CDC declared in 2013 was a serious threat to public health because of its resistance to multiple classes of antibiotics, okay? And the World Health Organization just released a report in 2017 declaring that Acinetobacter baumannii is at the top of the list for concerns for multi-drug resistant pathogens. Okay, so the project that we've developed at this point, and I'm in the process of coaxing NIH to believe that this is a fabulous project, we want to develop a bivalent mucosal vaccine where we target outer membrane proteins OMP-A and OMP-W. OMP-A, outer membrane protein A, and OMP-W, outer membrane protein W, have been proven in mouse experiments to be challenge experiments to be protective against a challenge with baumannii. Okay, and obviously we want to express these outer membrane proteins on the surface of the bacteria, and in this particular case, we want to target extracellular export of these outer membrane proteins. All right, the problem is that OMP-A has this annoying tendency to grab onto peptidoglycan and stabilize the outer membrane. So overexpression of OMP-A might be problematic for vesicles because it's holding onto the peptidoglycan. All right, so we've had to engineer a version of OMP-A in which that interaction doesn't happen anymore. And this is the way the vesicle delivery system might look. So we've got CVD910 SSB stabilized. Now CVD910 makes its own copy of OMP-A. That's OMP-A ST here in the green. So if we brought in OMP-A from Acinetobacter baumannii, we might be able to produce outer membrane vesicles here when we brought in a plasmid that expressed some sort of outer membrane vesicle protein, inducing protein, for example, Cli-A, or later on we'll talk about PAG-L. All right, but because the OMP-A gene may be coordinating with the peptidoglycan, the purple layer there, we may get low levels of expression of the vesicles. Okay, so what if we delete the OMP-A from Salmonella typhi and express OMP-A from Acinetobacter in its place? If we engineer that OMP-A from Acinetobacter so that it no longer interacts with the purple peptidoglycan layer, then we may get more vesicle formation if we bring in hypervesiculating proteins like Cli-A or Pag-L. All right, and here, this is the pièce de résistance. If you have delta AMP from ST, so that it's not making its own AMP, it's expressing foreign AMP from Acinetobacter instead, and outer membrane protein W from Acinetobacter baumannii, then you could get bivalent vesicle formation and delivery to the immune system. And this is what we're attempting to engineer. First thing we have to demonstrate is that we have surface expression of an outer membrane protein from Acinetobacter baumannii. And so we did surface labeling experiments with 910 expressing AB-AMP, hopefully on the surface, using antibodies specific for AMP, okay? So we targeted antibody against AMP, and then we had an Alexa 486 as a secondary fluorescent reporter, okay? And as you would expect, antibodies against AMP from baumannii do not cross-react with Salmonella Typhi AMP, so there's no fluorescence here. When we introduce our low copy number expression plasmid expressing altered AMP from AB so that it's been engineered not to interact with the peptidoglycan layer, we get very, very good expression on the surface, okay? However, we decided we wanted to do an experiment where we permeabilized the CVD910 expressing AB-AMP. So this allows antibody to get all the way into the periplasmic space. So the idea is, what is the maximum amount of AMP that we can detect, whether it's on the surface or buried inside the periplasm? And you notice the overlap between the green curve and the yellow curve. We're getting about 60% surface expression of AMP, which I thought was really quite good. And what really surprised me is when we did this Acinetobacter baumannii positive control, we get a mean fluorescence that's just about the same as CVD910 expressing AB-AMP. So this is a very encouraging slide because this is very encouraging data. We've actually been able to engineer a Typhi strain so that it looks like a baumannii strain on the outside because it's making a lot of Acinetobacter baumannii AMP. So this was very encouraging and shows clearly that when we, at least for AMP, when we express an outer membrane protein from baumannii and indeed is able to be transported to the surface. All right. So this slide gets a little complicated, but it shows clearly that to get AB-AMP to the surface that's fine, but if you can't export it in vesicles, then you have a problem. All right, now this is where I need to emphasize. Remember, Cli-A makes outer membrane vesicles, okay? It's hemolytic, and it has two functions. It's hemolytic, and if you express it at high levels, it makes outer membrane vesicles as well. So what we did here was we wanted to find out if we use Cli-A as an outer membrane vesicle system, inducing outer membrane vesicles, will the hemolytic activity of those Cli-A vesicles go up in various backgrounds, which I'll explain in a minute. The idea being that if the hemolytic activity goes up, and these 910 strains are also expressing AB-AMP, the inference is that the AMP is going out as well because the vesicle export is increasing. Okay, so I'm using Cli-A as a reporter here for function. Can you get AB-AMP-seeded vesicles off the surface of CVD910? All right, so if you look at lane 1 PBS control, there's no hemolytic activity because Cli-A isn't being made. CVD910 by itself, there's no Cli-A being hyperexpressed. If you bring in PSEC10 expressing Cli-A, you get an increase in hemolytic activity. Now, this is the 910 strain that I talked about, where the deletion of wild type OMP from S. typhi has been deleted from this strain. You bring in Cli-A, and in fact, the hemolytic activity does indeed go up. So if you take away the OMP gene altogether, you can make more vesicles. Here we have in lane 5, we have a carrier vaccine where the ST OMP is gone. We've integrated AB OMP into the Guabier locus, okay? So these are exponentially growing cells in this particular lane. We put in the Cli-A hyperexpression, and indeed, the hemolytic activity goes up even more. So when I saw that particular data from that lane, I realized OMP needs to be expressed in Typhi for Typhi to be able to efficiently make outer membrane vesicles. You have to have OMP present, okay? And then in lane 6, we integrated the AB-OMP into the RPOS locus, okay? You notice hemolytic activity went down. Remember, RPOS is induced in stationary phase, okay? These cells were harvested in exponential phase, and so the hemolytic activity went down. But if you harvest the cells in stationary phase, remember, all of these panels are corrected for cell density, so we use the same number of cells in these assays. The hemolytic activity is very high. And this was really surprising to me, because this is the first time that a foreign antigen, AB-OMP, expression in a Typhi strain can be shown to benefit the vaccine strain. I've never seen that before, because typically when you express foreign antigens, it's an energy drain, it's a carbon drain, and the vaccine gets no benefit from it. But in this particular case, I've managed to demonstrate that AB-OMP in place of ST-OMP actually benefits the cell, the vaccine strain, and it's also a vaccine strain. Okay? However, for vaccinating human beings, okay, hang in there, I'm almost done. For vaccinating human beings, the FDA tends to get nervous when you're overexpressing virulence proteins like hemolytic hemolysins like Cli-A. And so I needed a system for inducing outer membrane vesicles that was safe, non-hemolytic. And this is when we discovered PAG-L, which is an outer membrane protein. PAG-L is very special. It's a deacylase that converts lipid A from a highly reactogenic hexaacylated moiety to a detoxified pentaacylated. So instead of six acyl groups, there's only 5. And this is a less reactogenic form of lipid A, all right? And surprisingly, it was discovered in a 2016 report that overexpression of PAG-L causes outer membrane vesicle formation, all right? So we wanted to find out if PAG-L actually did induce outer membrane vesicle formation in our carrier vaccine, CVD910. Okay, so this is a test of function slide here. Again, we're using hemolytic activity again, but in this particular case, we're using chromosomally expressed Cli-A. So remember, there's a drop in copy number now because it's only in the chromosome, okay? 1 copy per cell. And we're just seeding the membrane with Cli-A so that if we can get vesicles to form, they will be hemolytic, all right? So in lane 1, PBS control, no hemolysis. Lane 2, CVD910, no hemolysis. CVD910, delta Guabier, OMC Cli-A, no hemolysis, because we're not making enough to make vesicles. We're just seeding the membrane with Cli-A as a phenotypic reporter. And then when we brought in PAG-L version 1, I'll get to that in a minute, we saw outstanding hemolytic activity. Then we saw a reduction in activity with version 2, and we saw the lowest amount of activity in version 3. So what is version 1, 2, and 3? We engineered the ribosome binding site in front of PAG-L so that version 1 is an optimized ribosome binding site, so we get lots of PAG-L. Version 2 is a less efficient ribosome binding site, so we get less PAG-L, less hemolytic activity. And version 3 was an unoptimized ribosome binding site, and we got the lowest expression of PAG-L and the lowest hemolytic activity. So this shows very clearly that PAG-L indeed, in an inducible fashion, is able to mediate hypervesiculation of outer membrane vesicles off of CBD910. And if those, instead of Cli-A, if they were carrying A-beyond-A, you'd be getting a beautiful delivery to the mucosal immune system. All right, so this is the almost last slide. Here's what our candidate bivalent CBD910 carrier vaccine against Baumannii might look like. We can integrate OMP-W into the Guavier locus. We can integrate OMP-A into the RPOS locus. If one of those was less immunogenic than the other, let's say OMP-W was less immunogenic, we could also integrate that into the HTRA locus, and we would have PAG-L on an SSB stabilized plasmid. Okay, so in summary, we think that attenuated S. typhi live vaccine strains represent a remarkably flexible platform for developing multivalent carrier vaccines. Okay, hopefully I've convinced you that a marriage between plasmid-based and chromosomal expression systems will be very efficient at expressing high levels of foreign antigens without over-attenuating the strain with subsequent loss of carrier vaccine immunogenicity. Achieve the balance, okay? I think I've shown reasonably clearly that foreign antigens expressed on the surface or exported are more immunogenic than cytoplasmically expressed proteins. Perhaps more importantly, if we can get them out to the surface, we've developed an inducible, not constitutive, outer membrane vesicle system for delivery, efficient delivery of these foreign antigens as vesicles to the mucosal immune system. And last but not least, if you remember the surface labeling experiment that we did, you noticed that I only got about 60% surface expression. So of course, I'm tweaking that right now to see if I can get 80% surface expression. In other words, I'm always trying to optimize this to the best I possibly can. Okay? And last but not least, I didn't do this by myself. We've got engineers, we've got immunologists, and we've got what I call the heavy hitters, which are the senior investigators at the Center for Vaccine Development that have kept me out of trouble more often than I care to admit. Thank you very much for your attention.
Thank you, Dr. Galen, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Let's get started. Our first question is your technology limited only to outer membrane protein antigens? What if you wish to target an antigen not normally exported to the outer membrane?
Okay, so we actually we presented some data around in the middle of the presentation where we were developing a vaccine against anthrax. And if you recall, PA83 is normally not exported to the surface, so it was cytoplasmically expressed. However, when we fused that to Cli-A so that we could move it out to the surface, it became efficiently exported to the surface. And when we tested those constructs in non-human primates, the exported version of PA83 became much more immunogenic. So I could use Cli-A to move antigens to the surface, or I might be able to identify in the future another carrier protein that can move things out to the surface.
All right, and we're actually going to only have time for one more question. Why have you only used an intranasal model for immunization using attenuated S. typhi carrier vaccines? Can't you use intradermal immunization or some other route?
Okay, so we can't use carrier vaccine. Live bacterial vaccine strains cannot be used in intradermal immunizations because they would be very reactogenic because of the lipopolysaccharide on the outside of the cell. With the advent of PAG-L, because PAG-L detoxifies lipid A, I still wouldn't use Typhi carrier vaccines for intradermal immunization, but I have the option now of purifying the outer membrane vesicles that are induced by PAG-L, which are less reactogenic, and I could use them in a heterologous prime boost for parenteral boosting with recombinant OMVs.
Okay, well, I would like to once again thank Dr. Galen for his presentation. I would also like to thank Labrus for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December of 2018. You will receive an email from Labrus letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.